Hi everyone, my name is Annie and I'm your Vice President. I'm going to be giving you a brief intro to bioinformatics. So first of all, what is bioinformatics? So bioinformatics is an interdisciplinary field of science, and it combines a bunch of different fields, as you can see by this Venn diagram, including biology, computer science, information engineering, mathematics, and statistics to analyze and interpret biological data. This has really important implications for discovery of new diseases and causes of those diseases, as well as the development of new drugs. So first, I'm going to give you a brief introduction to what we're going to be doing in this workshop. So we're going to be going over the Gene Expression Omnibus Dataset, or GEO, and how to extract useful information out of it with differential gene analysis. This is a very brief introduction, so there's obviously a lot more detail that you can look up on your own, but this is just a starting point for you. So the first step is to get familiar with the GEO database. So I would highly recommend going to the NCBI GEO database and just playing with it and seeing what's on there. And so just a brief introduction to what it is. GEO is an international public repository that archives and freely distributes microarray, next-generation sequencing, and other forms of high-throughput functional genomics data submitted by the research community. So this is just straight taken from their website. So the first thing I wanted to talk about was, how do you choose a good data set? There's all these data sets out there, but how do we choose the best one? Especially when you look at GEO, you might be overwhelmed. But I'm just going to give you 3 points that would make it a lot easier for you. So a good data set is one that, first of all, has a sizable amount of data for what you want to do with it. So the size depends on the context and the purpose of your research, but generally for our purposes in biomedical research, we want as many as we can get, and obviously for patient studies that might not necessarily be a lot. It may be 30 or 40 people, but if you want to do something very meaningful, you would want at least 100 patients. So since the samples are coming from these patients, we want to just take studies that we think would have the least room for error. So whatever you can find as the maximum. Another component of a good data set is being high quality. This includes being reliable, so that means that the study makes sense for the data we want, as well as the data being normalized and general outliers being removed, so it's not skewed for any reason. The normalization and the removal of outliers can be done by ourselves. It doesn't necessarily have to already be implemented within the database, so as long as it's not completely skewed, it could make for a good data set. So the next step would be looking for a data set on GEO. So this is kind of how the screen would look for the Gene Expression Omnibus. And if you already have a data set in mind, you can just go ahead and type in the name of the data set here and press the search button. Otherwise, you could even go to the search bar and type in what you're looking for specifically. For example, I could type in Alzheimer's and it would populate results with a bunch of related data sets. So this is the advanced search builder. Right in the search box with the arrow, you would type in Alzheimer's and you would end up with a bunch of different results. So the next step, once you find a data set that you think would work, so this one is GSE 118553. You will have a lot of different information on this accession display. So this includes the status, so when they made it public, the title of the study, the organism, the experiment type, and a summary of what the study actually accomplished. The important things here are circled in red because this gives you details about what exactly the researchers were studying. So as you can see here, they were looking at individuals with Alzheimer's disease and asymptomatic Alzheimer's disease and trying to find a connection. There was also a control group. So then, if you scroll down, it gives you more detail into the methods of their studies. And you can see the total number of people was pretty high. They had 27 control, 33 asymptomatic Alzheimer's disease patients, and 52 Alzheimer's disease subjects. And so it gives you a lot more details here as to what the genes that were associated with AD and some of the disturbances in various parts of the body that they observed. So reading this stuff has to do with the phenotypic data, and it's very important for you guys to be able to read through a lot of the study itself, because otherwise all the data analysis you're doing later makes no sense and has no meaning if you don't understand the study where it came from. So I would highly recommend looking through the phenotypic data before actually jumping into the analysis. So now let's talk a little bit about GO2R analysis. So we can use GO2R to compare two or more groups of samples to identify genes that are differentially expressed across multiple experimental conditions. So keep in mind, there are a few different forms that you will see when you click the Analyze button. You will see that there's a mini ML file, soft, or .txt. Mini-ML is a data exchange format optimized for microarray gene expression data, as well as many other high-throughput molecular abundance data. SOFT, which stands for Simple Omnibus Format in Text, is essentially what GEO uses as a data exchange format. An advantage of SOFT is that it's very simple and makes it suitable for parsing by virtually any text manipulating language. And finally, Mini-ML is basically just an XML format of SOFT. So for you guys, I would highly recommend just downloading the one with the .tar extension on the bottom. And if you have a Windows, make sure you unzip these files, otherwise you can't view the files. But if you have a Mac, you should automatically be able to view the files and load it into RStudio. So now you can press Analyze with GO2R. This allows you to analyze the data set. So you can either analyze on GO2R or RStudio or both, but this just has a built-in R script, so you have even less work to do when doing a preliminary analysis. The first step now is to define the groups. So as you can see, when you press Analyze with GO2R, this screen pops up, and it gives you a bunch of different samples here, and then the title of the samples, where in the brain these samples were taken from, gender, age, the subject, the tissue, and then the disease state. So because we have all these different factors, there's different ways we can define groups. We could look at just the brain, part of the brain, and see whether that related to a difference between the control and the Alzheimer's. Or, and what we can do is actually just look at the differences between the people who had Alzheimer's, who had asymptomatic Alzheimer's, and then the control groups. But keep in mind, you can only look at two groups at a time to get the volcano plot that we want later on and make some meaningful conclusions. So as you can see here, once you click Define Groups, you will see a pop-up that allows you to enter your group name. So just define it based on what you see here in the table. So if we want AD, which is Alzheimer's disease, just type in AD, and then you'll be able to select the groups you want. So here it's an example of what you could do. So for example, if you wanted to select the top 4, you could select those patients, those samples, and then type in AD, and then those would be highlighted a certain color to indicate that those were the defined groups. So for the purposes of saving time, I didn't define the entire data set. I just added about 30 samples per group. So before doing the analysis, the first thing you want to do is actually do a value distribution. So there are a bunch of different tabs here that you could look at GO2R, but the first one we wanted to do is actually value distribution. So this is just a backup to make sure that your data doesn't have any weird samples that might lead to odd results. After doing this, you can see that all of them look about the same in terms of the median and quartile signal ranges after being normalized. So the medians are just indicated by these lines here, and the different colors just indicate the different groups we defined. So for example, we did AD and control. And then at the bottom, you see the sample names so that you can remove any outliers if you see any before moving on to the next analysis part. Now you want to click top 50, go back to the GO2R tab and click top 50, 250, and then it'll start processing, which might take a little bit of time. So just be patient with that. Alternatively, if you think that it's taking way too long on Chrome, I would recommend trying it on a different browser, because I moved to Microsoft Edge and that worked a lot faster. So this is what, after you press that, your results might look like. Sorry, this is a little bit blurry, but basically you have different columns here. You can see that there's the ID of the sample, the adjusted p-value, and then a few other things. And another important thing is the log fold change, which we'll talk about later. And then gene symbol, and then gene title, which are just the different annotations about the gene. So what we want to do now is actually save the results. So either you could just look at this and then do some of the calculations on GO2R, Or you could take it further and then save these results to your computer and then do some analysis on RStudio. So click Save All Results, and then that will put it in a text file, and then make sure it's saved to the appropriate directory, the same directory that you are coding in RStudio. So now it's time to code. What we want to do is actually make a volcano plot. And a volcano plot is just a type of scatter plot that shows statistical significance, so the p-value, versus the magnitude of change, which is the fold change. And fold change is just basically the variance between the two groups. It gives you another indication of whether you have outliers. And so as you can see on this graph, there's the blue are the down-regulated, the red are the up-regulated, and the gray are the not significant ones. Our volcano plot will look a bit different, but the principles are the same, and the x and y axes will be very similar. Okay. So make sure that when you are running this code, you are using R 4.0.0 or 4.0.1, and use RStudio to make it easier for you guys. And then we're going to be using the enhanced Volcano package. So I'm just going to go over some of the code to do this, but I would highly recommend trying this on your own before jumping into this. The first step is to install using BIOS Manager, install enhanced Volcano, and then load the library. Sometimes this doesn't work, which was the case for me. So if that doesn't work, try installing it directly from GitHub, and you can do it like this. So now I want you to see if you can write the code on your own to make the plot based on the documentation, and then come back and check for your answers. You can do this using the question mark function. So basically, question mark function name, and then it gives you, that capability gives you the ability to learn more about the function, and you can do this with any function, but in this case, we want to learn more about enhanced Volcano. So once you've done that, come back to the video and see if you're correct. This is the code for how to make the enhanced Volcano plot. So the first thing we want to do is actually read our data that we saved. So I saved the data to a text file called allstext.txt, and I indicated that the header was true, meaning that there were titles in the header, so you don't want to include that when you're dealing with numerical data. I saved that into gene table, and look at how I saved a variable in. I have to use this arrow thing instead of an equal sign. Next, I want to actually make row names for the gene table so that we can label our plot. So I took the row names of the gene table, and I saved that from the gene table ID column. So now the ID column that we got from this text file are the row names of the gene table we're using. And this gene table is what we're going to be loading into Enhanced Volcano to actually make the plot. Now, these are just a few of the different parameters we have to feed into Enhanced Volcano to make the plot. So lab just stands for label, and then p cutoff is just the p-value you want to stop at, which is 0.05, which is generally what is considered significant. Then you have the x and the y scales, the labels you want to call them. And then just the point size, label size, and then the scale for your x-axis, and then the label size here. And this label size is for the legend. So once you do that, see if you can do this on your own, and then compare your results with this volcano plot. This is what it should look like. And then I want you to challenge yourself and see how you would interpret this. So if you go online, there's a lot of resources for you to learn more about exactly how to interpret a volcano plot. But anything outside these thresholds, so we have this dotted line here, so anything below this is not significant. That's why it says NS. But anything above this is blue. That means it's significant because of its p-value. And then anything red means that it's significant because of both its p-value and the log fold change. So these are labeled by their sample names. So these are things that we might want to consider taking out as outliers.